Hey guys, what's up? It's me Jay from CATCH25 Science Academy, and this time we've taken the first chapter from the biology HSC textbook. Yes, you've guessed it right, it is reproduction in plants. Now before we actually start the chapter, I'd like to revise some of the basic concepts of morphology of flowering plants, by which I mean the floral whorls of a typical flower, which are divided into basically two parts, the accessory whorls and the reproductive whorls. The accessory whorls consist of the outermost calyx, which consists of the sepals, then the corolla, which consists of the petals. Then we move on to the essential or the reproductive whorls, which are known as the androecium or the male reproductive whorl, and the gynoecium, which is the innermost and the female reproductive whorl. So moving on. So as the name suggests, reproduction in plants, reproduction is basically of two types, asexual and sexual. And right now we are only going to consider the sexual reproduction, and by which I mean that we are only going to consider the two whorls, the male reproductive whorl of the flower, which is known as the androecium, and the female reproductive whorl, that is known as the gynoecium. So after moving further, androecium consists of a singular unit known as the stamen, while the gynoecium consists of the single unit as carpel or pistil. Now further, stamen is divided into three parts: the filament, anther, and the connective. Now actually the main component of the stamen we consider as the anther, and further moving for female whorl or the gynoecium, which consists of the carpel or pistil, consists of the main component as ovule or integumented nucellus. Why do I say the name integumented nucellus? We'll study further. Now the anther further consists of something known as microsporangium, pollen chambers, pollen sac, or locules. That's four names. Then further with female whorl, the ovule or integumented nucellus consists of the megasporangium or the nucellus. Now the HSC students often confuse between the microspore mother cell and the megaspore mother cell, which forms the megasporangium and the microsporangium. So I'd suggest that instead of microspore mother cell as MMC, you try to remember as the PMC, the pollen mother cell, because that's comparatively much easier. Anyways, moving on. For the microspore mother cell, or I must say the MMC or the PMC, it undergoes meiosis, or in this case we must say microsporogenesis. As the name suggests, microspore, as you know, and genesis, it means production or the synthesis of it. So microsporogenesis or meiosis of one MMC forms a tetrad, or 4 microspore or 4 pollen grains, or a tetrad of male gametophytes. Now these 4 male gametophytes will undergo mitosis twice, and total of 8 male gametes is formed. Moving on with the female whorl, where megaspore mother cell undergoes meiosis, or in this case we must say megasporogenesis. As you know, genesis is synthesis or production. In the megasporogenesis, one MMC is going to form a tetrad of megaspore, not microspore, megaspore. Don't get confused between these. So anyways, out of these 4, 3 of them are going to get degenerated, and 1 is going to survive. To be more precise, the one which is near the chalazal end is going to survive, and this one leftover MMC is gonna undergo mitosis thrice. Now this is going to form one female gametophyte or one embryo sac, which in turn forms one female gamete. So don't write that one of this survives and forms one female gamete. That's wrong. It forms one female gametophyte, which forms the embryo sac, and which in turn forms one female gamete. Moving on with the male whorl. Male whorl, as I said, it consists of the anther, connective, and the filament. We are going to consider anther for right now. So now the anther consists of the anther in nature is actually bilobe, dithecous, and tetrasporangiate. Now what do I mean by this? Now consider my hands as the anther. Now if I open my hand, you can see how many palms are there. There are 2, right? So now that's the exact nature of the anther. Anther is bilobe, hence the name bilobe. Now tetrasporangiate. Each lobe consists of 2 microsporangia. So therefore in total there are 4 microsporangia, which gives the name tetrasporangiate. So now the leftover term, which I must say dithecous. At one time, both of the microsporangia in one palm, I must say, or one lobe of the anther, at one point of time, both of these microsporangia are going to fuse together and form one unit known as theca. And therefore, and because one lobe will consist of one theca, hence 2 thecus, which forms the name dithecous. Now moving further, The microsporangium consists of something known as sporogenous tissue, which in turn is formed due to the spores, or I must say the MMC or the PMC, which is diploid in nature. Now moving on to the layers of anther. In the layers of anther, first of all we come to the outermost layer, which is the epidermis. Then we come to the inner layer, which is known as the endothecium. Then comparatively a much thicker layer than both of these is known as the middle layer, and the innermost layer, which is known as the tapetum. Now tapetum has two major important functions. First is the nourishment of the PMC or the MMC, or you can also say the sporogenous tissue. The second function is the secretory nature of it. By secretory nature, I mean that it secretes something known as sporopollenin, which in turn forms the exine. Now the exine is something like, wait, let me show you. This is a pollen grain, and now you can see these crescent moon-like structures. These are something known as the exine, and the round structure which is being covered by the exine is known as the endine. Now you can see that this thing known as exine, this is the one which requires sporopollenin for its formation. And now the inner structure, like the inner ball or the sac-like structure, this is known as the entine, which is, you can see that it is divided into two parts. This one is known as the smaller part, or the generative cell, and the bigger part of it is known as the vegetative cell. Now the generative cell has the function that it generates the male gametes, and the vegetative cell is the one which provides with the nourishment for the pollen grain for its overall growth. Secondly, I must say that entine is made up of cellulose and pectin, and as you know that exine is made up of the sporopollenin. Now once the pollen grain reaches the stigma, now consider this as the stigma. Now once one pollen grain reaches here, the secretions of the stigma are going to be consumed by this pollen grain, and it is going to form an elongated tube-like structure, which is known as the pollen tube. Now how does this escape? So as you can see that the exine does not completely cover the entine. There is a free space here, and here. Now this free space between the exine and the entine is known as the germ pore, and from the germ pore the entine escapes and it runs down the style when it reaches the stigma. And with the pollen tube itself carries three major components, which consist of two male gametes and one tube nucleus. Moving on. Yeah. So in the female world, I must say, the ovule in this case, ovule is covered by two layers known as the outer integument and the inner integument. And now consider this as an ovule. So now my ring finger and the small or the pinky finger are locked, and now my central fingers are considered, you consider them as the inner integument, and my index fingers as the outer integument. Now the central part, which you can see is locked, is known as the nucellus. Hence the name I suggested earlier was the integumentum nucellus. Now once we reach the integumentum nucellus, and it is covered by these two integuments, the outer and the inner, the free space by which the two arms of the integuments are separated is known as the micropyle end. And the exact opposite of it, which we must say the bottom of the nucellus, is known as the chalaza. As you can recollect that the chalazal end, the microspore mother cell which is near the chalazal end is the one which is going to survive. Moving on, where we must say that So the chalaza is, you can consider these 2 points as the chalaza, and the chalaza is the one we can consider as the bottom of the nucellus, and below the chalaza lies the funicle. Now funicle is the thing which connects the ovule and the placenta, and in this case, like, we must say that if there is a case where funicle is absent, the ovule is known as the sessile ovule. So we continue after the sessile ovule. As we said that in megasporogenesis, a tetrad of megaspore is formed. Now out of which, as we said, in this ovule, the 3 of them which are near the micropyle end are going to get degenerated, and the one near the Chalaza end is going to survive. Now, as it undergoes mitosis 3 times, first it will form 2 nuclei, then 4, and in the third time it will form 8. Now there are 4 nuclei in the micropyle region, and then there are 4 in the Chalaza end. So now one from each of the pole is going to come in the center, and now 3 cells are formed in the micropyle end, out of which the central one is the female gamete, and these 2 in the sides of the female gamete are known as the synergids. Now in the Chalaza end, there are 3 cells known as the antipodal cells. Now we continue with the pollen grain and the stigma. So as the pollen tube elongates, it reaches the ovule. This process is known as the syphinogamy. And now there are mainly 3 types of syphinogamy, or 3 routes by which the pollen tube can perform syphinogamy. One is directly entering the micropyle end, which is also known as the porogamy. One of them is known as the mesogamy, which means through the integument to the micropyle end, and third is known as chalazogamy. From the Chalaza end, it reaches the micropyle end. Now once syphinogamy is performed and the pollen tube reaches the ovule, one of the synergid and the pollen tube both burst down, and since now the pollen tube is open, both of the male gametes enter the nucellus, or the central cell, we might say, and now one male gamete is going to fuse with the female gamete, and this process is known as syngamy. Now once syngamy is done, a thing known as a zygote is formed, which is later going to be known as the embryo. And now since we had 2 male gametes and one of them is fused with the female gamete, the other one which was haploid is going to fuse with the secondary nucleus, which is diploid in nature, and hence the process of their fusion is known as the triple fusion. And now once these both are fused, it is going to form the primary endosperm nucleus, and the thing which we once called the primary endosperm, which we once called the central cell, is now known as the primary endosperm cell. And of course the triple fusion causes for the formation of the primary endosperm nucleus, and the male and female fusion causes the zygote. Now post fertilization, zygote in future is going to turn into embryo, and the PEC, or the primary endosperm cell, is going to be known as the endosperm, and endosperm is the thing which is going to provide nourishment to this embryo. So that's all for this time, and wait, yes, that's all for this time. Hope you enjoyed this video. Until it's me, Jay Dave from CATCH25 Science Academy, signing off.